Since I see uh, there are very young people as well as I think 10th, 12th standard kids as well. So my first question is, are you familiar with, how familiar are you with chemistry? So like DNA, proteins, lipids, these things you have heard previously? Any? Okay, don't call me sir, please. My name is Roni. You can call me Roni. Okay, so what I'll be talking today is something called the protein folding problem. So let's first talk about what proteins are. So proteins are these molecules which are present inside cells. So I am hoping that you know what cells are. In case you don't, so cells are the fundamental building blocks of life. So what does this mean? This means that if you break an individual, a living organism, to its smallest component which is still living, you will come down to cells. And cells are mostly composed of three prime components. They are called lipids, nucleic acids, and proteins. So why have I listed these three names? So let's talk about what a cell has to do. A cell has to differentiate itself from its surroundings, so it needs a boundary of some sort. So these lipids are what is making these boundary. So the cell membrane is composed of lipids. Then what the cell needs to do is have some idea of what to do in case some situation presents itself. So it needs a set of instructions. The set of instructions is what is coded in nucleic acids. In most cases, it's DNA, deoxyribonucleic acid. And then what it needs is to do some functionality. So let's say it finds some chemical. What does it have to do with the chemical? It has to some sort of metabolize itself, metabolize the chemical, or it has to move from one region to another region. And all that functionality is carried out by proteins. So basically it needs some sort of demarcation, some sort of instructions, and then some sort of molecules to carry out the work. And proteins are the molecules which carry out most of the work done inside the cells. So what is a protein? A protein is composed of these things. Too much chemistry, don't worry about the structures, don't worry about the names. Basically, what you have to remember is there are some around 22 names here. So these 22 names in any order will compose a protein. So a protein may look like C, G, P, Y, F, M, L in any sort of order. And so here these things are classified based on some chemical properties. So the first classification here says that amino acids with electrically charged side chains. So what do charges do? Are you familiar with positive charges, negative charges, right? So if I have a positive charge and a positive charge and I bring them together, what is going to happen? They will repel. Okay. If I do same with negative and negative, they will repel. Positive, negative, they are going to attract. So to put simply, like charges repel, unlike charges attract. Okay, keep that in mind. Now what we have again here, it is saying amino acids with hydrophobic side chains. So what does hydrophobic mean? Hydrophobic means something that does not like water. For example, oil. Let's say I put two oil droplets in water. What is going to happen? They will float in the surface. What if they come together? What will happen? They will join, they will coalesce. Okay, so basically hydrophobic side chains, if I have some let's say some whatever this name, phenylalanine. Let's say I put two phenylalanines in water, they will come together. Okay, also keep that in mind. Now this says polar, uncharged, whatever side chains. So what does polar mean? Polar basically means that these amino acids will like to be with water. So these charged side chains and these polar side chains like to stay with water. These are listed as some special cases. Why are they special? This one is special because this, sorry, who is asking the question? Please raise your hand and ask the question. No, ask. Who was the one who asked? I didn't hear. That's why I'm asking again. There was some question. Huh. Okay. No. These are parts of proteins. So proteins are different from DNA. DNA is a different molecule in of itself. These proteins will be made from the instructions given by the DNA, let's say. So that is cytosine. ATGC, you are saying the DNA is made of ATGC. So that C there is cytosine, not cysteine. Similar names, but different chemicals. Okay. So this one is special here because this contains sulfur. The cytosine one doesn't contain sulfur. It's just nitrogen, whatever, phosphorus. So this one contains sulfur. The glycine doesn't have a distinct side chain, and the proline is different because the proline sort of folds upon itself. Does not matter. Some chemistry. These have some sort of special properties of their own. In any case, what we figured out was that we have some charged interactions, we have some hydrophobic interactions, and we have some water and charge. So basically some degree of hydrophilic interactions as well. Why is this important? So in most of the cases, when proteins have to perform some of their functions, they need some sort of a structure. Now the protein folding problem is like this. Let's say I give you a string, a thread, okay, let's say a cotton thread, and I tell you that this thread has to make a specific sort of embroidery pattern by itself. That is very difficult to do. Something which self-organizes into a very distinct shape every time is a very difficult physical problem to address. Okay, so this is like saying I will ask all of you to stand in a queue, and you will be in the same position of the queue every time. Okay, so this is the sort of complexity we are dealing with here. But the physical properties which you discussed here help the protein to fold. So what will happen is in the protein sequence, in the thread, where there are these individual amino acids one after other, they will come together based on their chemical properties. So if there are one positive and one negative charge, they will come together. If there are hydrophobic groups, they will come together, and this will lead to some degree of organization in that thread. And what will happen is this thread will gradually organize into higher order structures to give us the final protein structure. So what do I mean by that? So we'll look at some sort of structural elements of proteins. So if I take a protein, the protein sequence, it will first fold into smaller portions like this. So this is the thread. The thread can fold into this spring like things or into this sheet like things based on their chemical properties. Okay? And these smaller spring or sheet like things can now fold again. They can also interact with other sheets, other helices. So these things are called helix, right? So this is a right-handed helix, and these helices will again come together to form a full protein structure. And then multiple proteins can come together again to form even more complicated structures. So what I will show here is some proteins. So I have this. So how many of you have aquariums at home? Do you have aquariums at home? Anyone? Does anyone have aquarium at home? Okay, no one. But have you seen these glowing fishes? So these fishes, you like basically shine some UV or something on them, they will glow in the dark, right? So these fishes glow because of some protein like this. This is called a green fluorescent protein. So you can see that this protein is made up of multiple sheets. There are some helices inside. There is some some random coils or whatever. So all those helices and the sheets I mentioned, right? Those will come together to give you this nice looking protein. Another example which I have loaded is, so this one is actually a protein which you probably have heard of, hemoglobin. You know hemoglobin? What does hemoglobin do?
Oxygen to the cell.
Correct. So hemoglobin is the protein which carries oxygen to various parts of the body, right? So this coloring is based on hemoglobin subunits, doesn't matter. This thing is the part where the iron, the heme in the hemoglobin sits, and this is where the oxygen will come and bind. Okay? So this is the hemoglobin protein, which is very important for life. So as you can see, the structural elements, they sort of come together to give you this nice protein structure. And now, like, the question is, okay, I have told you what the problem is, I have told you what protein structures are. So let's try to do an experiment. There was some chalk here. So let's say I have this is some thread which I was talking about, and these are the amino acids on the thread. Okay. Now let's say in this amino acids, I put some A here, some B in the middle, and some C at the end. Okay. Is this clear? So what I will say is, this A will have some color. If A and B come together, there will be some other color. If B and C come together, there is some other color. And if A and C come together, there is some other color. Okay. So A, AB, BC, and AC. So, I mean, ABC all have their individual colors, together they will have some other color. Okay. So what will happen if A and B are together? You will see only B. If B and C are together, you will see only C. If A and C are together, you will see only the color of C. So this is how the color pattern will look. Is this clear? I am saying A, B and C individually have the separate colors. If A and B are together, you will see only the color of, you will only see the color of A. If A and B are together, you will only see the color of A. If B and C are together, you will only see the color of C. If A and C are together, you will only see the color of C. So let's say this thing folds, okay, such that A and B will come together first, and then A and C will come together, B and C will come together, and then A and C will come together. Okay? This will happen first, this will happen after, this will happen at the end. So now what I am asking you to do is, I will have you all are familiar with graphs, I am assuming. Okay? This here is time. I am measuring the time and I am trying to see colors of A, B and C. So what do you think will happen first when nothing is close to anything? You will see all the colors. You will see A, B and C, correct? Is this clear? Okay. So if I am measuring intensities of A, B and C, all of them will be high. So let's say this is A, this is B, this is C. Now gradually when A and B come together, we should only see A and B will die out. So A will increase, B will decrease, and then you will see now B and C are coming together. So B will decrease even more, and then gradually C will increase, and finally when A and C are together, A will go down, C will increase. So this will continue. You should see something like this. Okay? And is this understandable? Like the colors should change based on what things are sitting together closer to one another. So this is how actually you can conduct some experiments. I will show some experimental data. So you actually did some experiment, right? So you said that how the color pattern should look, and you can actually see that the colors, so this red color like sort of increases, sort of then dies down, the other color comes up, and finally all the colors come down and you can only see this green color. So what I did in this sort of thought experiment, you can also conduct experimentally and see fluorescence and stuff like that. So you can actually see proteins folding in real time. Okay. So more or less this is the portion of the talk. Now what I will do is, I have made some models for your beta sheets, and I will give you some pipe cleaners. So you can make alpha helices, I mean you can make helices out of this. The way to do it is, you hold this pipe cleaner with your thumb like this, and then coil it around your finger like this, okay? And this will become a nice helix, nice spring, and you will also get some copper wires like this. You can fold this pipe cleaner around the copper wire, and then carefully, seems I'm not strong enough to pierce the cardboard, right? Okay, you can do this, I can't do this. Basically you sort of pierce the cardboard through, and you can organize these helices and the beta sheets together in some fashion to create a protein of your liking, okay? So we will slowly distribute some of these helices and this copper wires and pipe cleaners, beta sheets, whatever, and you can construct a protein of your choice. Okay, if any questions you have, please do ask, otherwise I'm done with my talk.